Good luck, Dr. Restrepo.
Hi, Izzy. How are you?
Doing good. How's everything on your end?
Oh, good. Let me get my. Can you speak again?
Sure. Can you hear me
Okay, good. Yeah,
Awesome.
I can hear you now. Okay, good. Thank you. Let me check the sharing. How do I share my? It doesn't allow to share the screen.
Yeah, Izzy, my button is not highlighted either, so.
There we go. Okay, let me see if I can share. Let me see, screen 2, share, and I am going to do here. Oh, I'm sharing the wrong screen. Stop sharing. Let me see. Okay, let me see if I can share. Okay, can you see the presentation now?
Yes.
Yes.
Yeah.
Okay. Okay, good. So I am going to unshare while we get started. And then I'll know that that's what I need to do.
Sounds good.
Oops. So it is 11:30. Has Belinda joined? No.
Not that I see.
Okay,
Yes.
so let's wait a few minutes to see if Belinda joins.
Hello,
Sure.
Dr. Restrepo. This is Belinda Medrano.
I'm thinking of, hi Belinda, I see you. I'm thinking of Belinda Reiniger.
Okay, great. Thank you.
Yeah.
It's an early join. I wasn't sure if I could help in any way.
Yes. No, thank you. Thank you, Belinda.
Can you all hear me?
Yes.
Dr. Reinecker and Fisher Rock and McCormick are next door at the hospital at a meeting. They said they'd be back, so it's here ready for them whenever they come back in.
Oh, okay. Okay. Should I wait for them or do I get started since it's 11:30? What do you all suggest?
Hi, Blanca. Dr. Restrepo, I was going to do a quick introduction and whenever you want.
Okay, good.
Wonderful to see everyone. Thank you for joining us today. It's our pleasure to introduce our colleague and also our anniversary speaker series presenter today. So Dr. Blanca Restrepo is an associate professor of epidemiology at UT Health School Public Health in Brownsville. She has over 75 publications in peer-reviewed journals, several chapters that she's contributed to, and her work is very unique with a binational approach, as well as in informing the understanding of health equity, social justice, as well as health disparities in TB and other chronic illnesses. So Dr. Restrepo, welcome, and she will be presenting to us today on the elderly, diabetes, and tuberculosis, lessons from studying these vulnerable populations. Thank you very much and welcome, Dr. Restrepo.
Okay, thank you, Dr. Valerio. Thank you for that introduction. It is a pleasure to be here. So as Dr. Valerio indicated, so it's a pleasure to celebrate the 20th anniversary. I joined the school actually 20 years ago in October of 2001. So I've been pretty much from the very beginning, and I started at the school as a postdoc, and the school has provided me this environment for growth that I want to share with you today. So there's been a lot of things that have happened in 20 years, and I'll do my best to try to synthesize the most, what I thought are the key highlights. So I'm gonna share my screen. Okay, so I assume you all can see my screen. And with that, let's see. Okay, so this is gonna be the structure of my seminar. I am a specialist on tuberculosis and diabetes mellitus, and sometimes I abbreviate it DM. And I'm going to provide a historical perspective on the association between these two diseases. Then our epidemiology findings, which were in a way groundbreaking, and what public health implications these findings have had historically and may have prospectively with the studies that we're currently doing. Then I'm going to very briefly touch on some of the biological findings that complement this epidemiology. Then I'm going to skip into a new but related chapter of studying tuberculosis in the elderly. Why study the elderly? What are the unexpected epidemiological observations we have made? And what are the clinical implications? Then I'm just going to touch one slide of how we're moving into TB diagnostics. Throughout I've prepared a special seminar today for this occasion because I want to highlight the work of our students, our collaborators as SPH faculty, community service. So I may not mention everybody, but I have tried to do my best to highlight those who have participated. So Mycobacterium tuberculosis is a bacterium, and it is the etiological agent of a very serious pulmonary disease called tuberculosis. In 2019, there were 100 million new cases of tuberculosis worldwide, and there were 1.4 million deaths due to tuberculosis. To put it into perspective, this is the #1 microbial killer worldwide for many years, except last year, COVID got ahead of TB. But TB has been with us for centuries. So how does tuberculosis occur? So you have somebody with pulmonary tuberculosis, and this is very similar to COVID. So it is an airborne infection, and everybody who is a close contact has a risk of becoming infected or sick. So every time there's a TB case, there's a contact investigation, just as there is with COVID. And so one of the most intriguing questions about tuberculosis is you can have a very close contact of a TB patient, and they can even be the sexual partner. And despite that, only 60% of the people become infected. And they become infected based on a tuberculin skin test that everybody has had a tuberculin skin test or an interferon gamma release assay. And that's how we know that somebody has a latent TB infection. And then among those with a latent TB infection, only 10% in their lifetime will progress to an active tuberculosis disease. And this is a very serious infection. but it can be cured with antibiotics. However, there can be complications, such as the delayed clearance of the bacteria, or the person thinks that they're cured and there's a relapse or a reinfection, or there's even death. So what determines the fate of somebody who is exposed to the bug? And that's going to be determined a lot, but what are the characteristics of this close contact? So anything that compromises your immune response is going to increase the risk of tuberculosis. So the biggest risk factor for tuberculosis at an individual level is HIV/AIDS. It increases the risk of tuberculosis by 20-fold. But today we're going to talk about diabetes. It increases the risk by 3-fold, and old age 1.5-fold. We're also going to talk about how this host may be even protected. So we're going to talk about the BCG vaccine and how it may reduce the risk of tuberculosis. It's known in children, but we're going to see a surprise in the elderly. So let's focus on diabetes. So diabetes is, quote unquote, a re-emerging risk factor for tuberculosis. And why re-emerging? Because if we look at the earlier part of the past century, the reports on diabetes and tuberculosis were fairly common in the literature. These are seminal studies that I'm pointing out here. And then it pretty much disappeared. What happened? Where there were several groundbreaking findings, insulin was discovered to treat diabetes, antibiotics were discovered to treat tuberculosis. So that the literature on TB diabetes pretty much disappeared. Then in the 1980s, we had the emergence of the HIV pandemic, and that really took front and center stage in TB epidemiology because it hit HIV patients are incredibly susceptible to tuberculosis. But in the meantime, we had here an increase in obesity and the diabetes pandemic coming up, and therefore an increase in the prevalence of TB diabetes. So we were right front and center in 2006. We had our first publications of TB diabetes. In fact, some of those first publications took years, more than 1 year to get published because the editors kept saying, there's nobody to review this, and this is something from the 1950s. And now you can say there's been an explosion of publications on the topic. So there's an interesting overlap between TB and diabetes. And what I did is I went to the literature and I took the countries where TB is most prevalent. There are 22 countries considered high burden by the World Health Organization. And then there are 10 countries with the highest number of diabetes patients worldwide. And if you put both of them together, 7 of these 10 countries with more diabetes are also countries with high burden for tuberculosis. So we've really got a quote unquote crisis of diabetes in low and middle income countries where tuberculosis is also endemic. And I claim that the Texas-Mexico border where we live is like a test tube to study what happens in the rest of the world. So this is our study site. It's in the, I have a very unique binational tuberculosis program, and I enroll participants on both sides of the border. And so if we look at the characteristics of this population, the incidence of tuberculosis on the Mexican border is much higher than the incidence in Mexico. Same thing in Texas. We've got a lot more TB here in the border compared to the rest of the US. In our border region, we also have a higher prevalence of diabetes, but in our border region, we have less HIV. So those are the characteristics. And in studies done by 2 graduate students, Jennifer Curry and Basant Abdelberry, an interesting finding contemporary to what we're seeing right now with the massive immigration is that 1 in every 3 TB patients with tuberculosis in the border are actually immigrants. They are not born in the border. So this is one more consequence of the immigration challenges we have right now in our community. In addition to the border, I've conducted studies in other parts of Mexico. Veracruz is not highlighted here, but I am going to present some data from Chiapas. This is the another border region for Mexico. It's one of the poorest states in Mexico. I've also have a very interesting collaboration with colleagues in South Africa where we're comparing South African colored versus Hispanics. And so the studies that I've done over the 20 years are basically of two types. They're retrospective epidemiological studies. So an advantage of studying TB is that TB is a reportable disease by law. So there are data sets available from the health departments for TB surveillance purposes. So the advantage of these data sets is that the number of people that we study are in the thousands, and they give us very valuable information. A disadvantage is that retrospective data is not always perfect. There's missing data. And then we're also limited to only studying people with active tuberculosis. On the other hand, these retrospective studies are the backbone to provide hypotheses that lead us now to study prospective epidemiological studies where we enroll participants, draw blood, confirm whether they have diabetes, what else is going on. And so here we study controls from the community, we study close contacts of TB patients, and we also study TB patients. So these are studies done in a much smaller number of patients, but they're very finely characterized by us. So I'm going to start by telling you the overview of what are the statistics on that association between diabetes and tuberculosis in the Texas-Mexico border. So first, the definition, what is diabetes? Diabetes is based on the American Diabetes Association definitions that have evolved over the years. And when we talk about diabetes, 98% of our diabetes patients have type 2 diabetes. And so diabetes can be defined as anyone with self-reported diabetes, and that's usually the basis when we do those retrospective huge studies. It's a lot of self-reported diabetes. But in our prospective studies, we also confirm diabetes based on hyperglycemia or chronic hyperglycemia. So using these definitions, one of the first questions is, what's the prevalence of confirmed diabetes? And this is when we started our field studies. And I want to acknowledge I was a postdoc at this time working with Dr. Fisher-Hawk and Dr. McCormick, who were my mentors for how to conduct field studies. And with Dr. Hossein Rahbar, who is a faculty member in Houston, who is like my teacher for the epidemiology that I know today. And so what we found both in Mexico and in Texas was that the prevalence of diabetes among TB patients, shown here in red, is significantly higher than the prevalence of diabetes in the corresponding community. These differences are significant, and the estimated relative risk of tuberculosis in someone who has diabetes is about threefold. So someone with diabetes has 2.7 to threefold higher risk of developing tuberculosis to compare to somebody without diabetes. As a reference here, I put HIV. HIV is 20-fold or more. So again, HIV is a huge risk factor. So having a risk of threefold, does it really matter? And the answer is yes. So we did these studies on the population attributable fraction. This is a very useful measure in public health. And what we do is the following. We find what is the incidence of tuberculosis per 100,000 in TB patients without diabetes. And this is what we find in our community. And then what is the total incidence of tuberculosis among everybody, with or without diabetes. And so if we cut here and say, this is the excess, this is the amount of TB due to diabetes, we conclude that nearly 1/3 of all tuberculosis in our community is due to diabetes. If we would eliminate diabetes, we would have 30% fewer TB patients. This is in contrast at the community level for HIV, which only contributes 3 to 6%. So even though at the individual level, someone with HIV has a huge risk of TB, at a community level, diabetes is a lot more important in our community. I want to highlight also work from another student, Hiba Rashak. She's originally from Iraq. She's back in Iraq now. And Basant Abdelberry, they did a study in Chiapas. As I showed you, Chiapas is on the other side of the border of Mexico, the poorest counties. Chiapas has huge indigenous presence. And so Hiba did an interesting analysis where she found that in communities where there's a predominant indigenous presence, diabetes, shown here in this lighter blue, is not really that important. What really matters in these communities is undernutrition. And as you have less and less indigenous presence, then yes, diabetes is important. The point being that worldwide, undernutrition is also a huge risk factor for tuberculosis. So it is important in every community to establish what are the priorities, where do we put our dollars in a community, and it's important to know the epidemiology in order to make these decisions. So what are the public health implications of these findings? So in TB clinics, well, TB clinics are there to diagnose TB, to manage TB. But with our findings, TB clinics have become a strategic site to screen and diagnose new diabetes. And so this is already established for TB patients, and our work has had an impact worldwide. Our work in this community has had an impact where the World Health Organization has endorsed that TB clinics, every newly diagnosed TB patient who is an adult should be screened for diabetes. And we find that the percent of diabetes patients who were not aware they had diabetes is 18% in Texas, 36% in Mexico among TB patients. What about the TB contacts? Well, this is more challenging because for every TB patient, there's at least 2, 3, 4, 10, 20 contacts. So the clinics could not go ahead and screen everybody. They're not going to have the budget to do that. However, what our findings show is that among TB contacts, no matter where you are, including South Africa, there's a high proportion of diabetes patients who did not know that they were diabetes. In fact, in South Africa, nearly half of the diabetes patients were not aware that they have diabetes. So we also find what are the characteristics of newly diagnosed diabetes patients versus those who already knew they had diabetes. They're more likely to be males, which is great because males usually don't go to the doctor. So this is an opportunity for males to find out that they have diabetes. They're usually younger than people who know they have diabetes. They have lower HbA1c. They have less diabetes complications. So you're catching diabetes at an earlier stage before the irreversible complications of diabetes. So this has a huge potential, but it's not going on right now. So I'm going to show you in the next slide some studies from students where we're working for models of how can we convince the world, let's not screen just among TB patients. Let's also screen for TB contacts. So I'm going to show you some data to support that. What is the role of the diabetes clinics? So the diabetes clinics have now become also a hub to screen for active tuberculosis, particularly in TB endemic countries, and this is endorsed by the World Health Organization. However, should we be screening for latent TB infection everybody who has diabetes? And the answer is no per WHO guidelines. What happens is that this is the math. We have about 9% of adults worldwide have diabetes. 25% of them are going to have latent TB infection, but only a fraction of them are going to have TB. So just the epidemiology alone is not sufficient for us to go ahead and screen for latent TB infection among all the diabetics. So one of the issues is we need to know how do we predict better who are the diabetics who are going to end up here with TB. And besides the epidemiology, we need to do biological sciences studies. So I'm going to show you a little bit of that. And then what would be the impact in general clinics? And this really comes in with COVID. So what is the impact of COVID-19 on TB? And so there are models now showing that with the COVID-19 pandemic, there's been problems at the programmatic level in the TB clinics, and we're going to have an excess of TB and excess of TB deaths due to COVID. But is there a synergy at the biological level between someone who has COVID and coming down with TB? And so we've done some really interesting observations at our field site. We have noticed patients who report having a COVID-19 episode, and they survive the episode, but they remain with a cough that doesn't go away. Then that cough migrates into a productive cough, and then they're diagnosed with tuberculosis. When we go back to see these COVID-19 cases with TB, what we find is that a lot of them have a history of chronic diabetes that is poorly controlled. So we hypothesize that these individuals probably had a latent TB infection. They had a chronic diabetes, which put them at risk for TB. But when they developed COVID-19, this really shifted the scale, and now they really moved into TB. So we're going to study this further with one of our studies, an MPH student, Liz Calles Cabanillas. So she's going to help us untangle these observations. So now I'm going to tell you about our studies among TB contacts and how we can screen, how we propose to screen for diabetes. So the first study is done by an undergrad student at UTRGV, Javier Rios, in collaboration with Dr. Juan Carlos Lopez Alvarenga from UTRGV. And our question was, how can we identify individuals who have a high probability of having diabetes, but without doing blood testing? So how can we stratify? So the strategy was to design a model to predict diabetes using non-invasive methods like anthropometric measurements and clinical history. So what we did specifically was we did a cross-sectional nested design using the NHANES 2017-2018 survey. And so this has 9,254 people. And after a lot of models, we selected a model that had the highest prediction for identifying new diabetes patients. Particularly, this one had an area under the curve of 0.783, so with 1 being perfect. And so we identified 3 variables that were good predictors: a family history of diabetes weighted 10 times 10, plus the body mass index of the individual, plus the age. And so here's what we found. So we had a score of 70, which would be recommended for most clinics. So this is the age of the individuals. This is our model, the score of our model. And anyone shown here in blue is people who do not have diabetes, and in red are people with diabetes. So if we say we're going to screen everybody with a score higher than 70, we're going to detect 90% of those with diabetes. And so that's what we aimed to do. So this is the model based on a survey from the entire United States, the NHANES. So we said, okay, well, let's test our model with people from the Texas-Mexico border. This is called a replication cohort. In this model, we use people who were TB contacts and community controls enrolled at my field site. And so what did we find? We had 586 individuals in this replication cohort. And if we if we're a clinic with more resources and we say, well, let's do a cutoff of 70. So let's do anyone that we're going to screen has a score of 70 and higher. So that is, somebody comes to the clinic, we ask for their family history of diabetes. If it is a yes, then they get 10 points. What is your body mass index? Whatever it is, you add that up. What is your age? If you add that up. If adding that up for you is more than 70, that means you're going to be screened with blood. Okay, you're going to have a blood test done. So with this cutoff, we would be screening 409 individuals, which is 70% of all the people in our cohort. And we would find that 56 of 59 new diabetics would test positive. We would detect 95% of the diabetics if we would do that. And so the savings would be 30% of everybody tested. That means we only had to screen 70% of the people in order to detect 95% of the diabetics. If we have a clinic with less resources, then we shift this line up, and that means we're going to test less people, we're going to find less sensitivity, but we're going to save more money. And so that looks very promising. We're in the processes of publishing these observations. And then we have with another student, Robert McGowan in Austin, and with help from Shelton Brown in Austin, who is a health economist, we did a cost-effective analysis. So is it cost-effective to expand TB contact investigations to screen for diabetes? So the methods, and I have to give a lot of credit to Robbie, he's a tremendous mathematician. So Robbie did a Markov chain model that simulates the progression of somebody with diabetes over a 45-year period. So what happens from the moment of diabetes diagnosis? What happens? What are the chances of complications? What are the chances of death? And what Robbie did is he compared the costs and the benefits of screening for diabetes and detecting diabetes earlier compared to not screening. This model is based on another diabetes simulation model created at the Centers for Disease Control with RTI. And it's and Robbie complemented from related literature from other sources. And we fed into the model demographic and epidemiological data from Hispanics, particularly from our Texas-Mexico border region. What Robbie found was that the cost of screening for TB for diabetes was between $15,000 and $27,000 per quality adjusted life year added. And I am not a health economist, but what Shelton says is that generally interventions that cost less than $50,000 per quality added are considered cost effective. So this is really within that cost effective range. What Robbie also found is that when the new diabetes diagnosis is followed by someone who does a successful management, so this person does a lifestyle change, takes their anti-diabetes medication, this leads to a 17% reduced risk of all diabetes complications and increased survival. So the conclusion from Robbie's model is that screening TB contacts for diabetes is cost effective, improves quality of life, and increases lifespan. So we're also in the process of publishing this, and we hope that this has an impact worldwide in the TB clinics. So I'm now going to shift focus to the biological basis of why diabetes predisposes to tuberculosis. So I know this is a very complex graph, but basically to show that when we consume something sweet, and there's a lot of glucose in our blood, so glucose would normally have this pathway to generate energy or to serve as substrates for who we are. When there's an excess of glucose, we start to have a lot of what we call these shunt pathways or alternative pathways. And this is not good. It leads to reactive oxidative species, in other words, oxidation, which is not good. And this leads to inflammation, a chronic inflammation, which is not good either. So all of these leads to what we believe is the reasons for why somebody with a chronic history of diabetes is more likely to develop tuberculosis. So we did some studies to understand this a little bit better. And so the studies were done in people who are TB naive, that is, they have never been exposed to TB, or people who are contacts of a closed TB case, or people who have progressed to tuberculosis. And so we asked, what is the impact of having diabetes at any of these stages and the ability in vitro of their cells to contain the growth of the mycobacteria? And so these are studies where another student, Diana Gomez, participated. And what we do is that we enroll participants at the field, we draw blood, and we enroll participants with and without diabetes. We isolate their white blood cells, and particularly monocytes and lymphocytes. They're all called mononuclear cells. We take these cells, we incubate them in vitro with Mycobacterium tuberculosis, and we check what is the response of diabetics versus non-diabetics. And so what we find is we can look at phagocytosis, which is the process of engulfing the bacteria. And then we look, what's the fate, the intracellular fate of the bacteria? Is the bacterium killed? Or does the bacteria feel at home and start replicating inside the macrophage? So what we find when we study monocytes from TB naive individuals is that if they are diabetic, they've got a reduced capacity to phagocytosis of MTB. And we've done a number of studies where we find they also have reduced capacity for phagocytosis of other pathogens. However, when we study TB contacts, now these TB contacts are going to have antibodies to the bacterium, and those antibodies are going to help with phagocytosis. So when we study TB contacts, we no longer see a defect in diabetes. But what we find is that both with monocytes or with mononuclear cells, when we have diabetics, we have an increased number of colony forming units, which means more bacterial growth inside the cells from people with diabetes. So this is very cool because we know that from the epidemiology, people who have diabetes have a higher risk of infections, including a higher risk of tuberculosis. And what we're seeing here in vitro in the lab is that we can replicate that. So we are now studying, now that we have found this, we are now doing studies in vitro, manipulating conditions to understand what is it about those diabetic individuals that compromises their ability to kill the bacteria. So these studies that I show you here are, we enroll the participants and right away, within 24 hours, we're incubating their cells with the bacteria because we want to keep the in vivo conditions as close as possible to the in vitro in the lab. But what happens if we take these monocytes and we differentiate them to macrophages, which are other cells that we have in our body? That takes 7 days. So we have a monocyte-derived macrophage, and then we add the bacteria. Will we lose that capacity to see these differences by diabetes status? And what we find is that in people without tuberculosis, people with high blood glucose, with diabetes, still have higher colony-forming units, although it is not as notable. What we find is people with a chronic history of diabetes will have significantly more bacterial growth. What we find also is if the person now has diabetes, there's more mycobacterial growth, which makes sense. This person developed tuberculosis, they cannot control mycobacterial growth, and we're seeing the same thing in vitro. And now having diabetes doesn't really matter. So this just shows the complexity of how to study in vitro the mechanisms by which diabetes makes someone more susceptible to TB, and how it can vary depending on whether someone is TB naive, is a TB contact, or is a TB patient. And so our working model based on our observations, I haven't told you all the basic laboratory findings, is the following. So let's say we have somebody here who is exposed to tuberculosis, and if they do not have diabetes, if they're an immunocompetent host, they're going to be more likely to contain the growth of the bacteria or even eliminate it. But if they are diabetic, they're going to be more likely to have mycobacterial growth. Then adaptive immunity kicks in, which is T cells and B lymphocytes and antibodies. And so they're going to be able, for the most part, to contain the growth of the bacteria, but not eliminate it. So people who have diabetes are more likely to have a latent TB infection. However, at some point, if that diabetes becomes chronic and poorly controlled, then it really increases the chances of losing that ability to contain the growth of the bacteria grows, and you develop tuberculosis. Not only that, but somebody with diabetes will also have more adverse TB treatment outcomes. For example, they're going to have higher mycobacterial burden. They're going to be more likely to fail treatment, to have relapses, or to die from tuberculosis. So we're in the process of understanding what are the detailed pathways that are going here to see if we can correct the deficiencies of somebody with tuberculosis. And this area of research is called host-directed therapy. That is, how can, if somebody develops TB, not just give them antibiotics, give them something to boost their immune response in the appropriate way to help those antibiotics and to help clear the bacterium. So I'm going to shift now to studies in the elderly Hispanics. And so why study TB in the elderly? So the global population is aging, and according to the US Census Bureau, in 2015, we had 8.5% of the world population over the age of 65. And we're expecting that that is going to duplicate by 2050 to 17%. And so there's an increasing, as we have an increasing age, there's an increased risk for TB disease. So we see here higher age groups in the number of cases per 100,000. And you can see in blue, this is all individuals, and it's particularly marked among males, higher risk for tuberculosis. But older age not only increases the risk for TB, more seriously, it increases the risk of death from TB. And this is in the literature between 6 to 8-fold. And this is shown also by our students. Basant Abdelberry showed in a multivariable analysis that individuals who were 65 or older had more than 5 times the risk of dying from TB compared to people who were younger. And then one of our current students, Belinda Medrano, she's got an MPH and she's doing her dissertation right now, and she's studying the elderly. And what she finds is that compared to the young people, as you get older, there's an increased odds of death. And it is like a dose response, with the highest among the oldest people. In the literature, about 20 to 50% of the elderly individuals who have TB will die during the course of TB. So this is a very serious disease in the elderly. So why are the elderly at higher risk of tuberculosis? So first, they had a higher exposure of TB to Mycobacterium tuberculosis as children. This is called a cohort effect, because TB was more prevalent years ago. So we've got among the elderly a large reservoir of people with latent MTB infection and with potential risk of reactivation TB, although I may argue that they may not necessarily be at such high risk. So this is a topic for another presentation. So another reason why the elderly are at higher risk of TB is because the elderly tend to have compromised immunity, and that will put them at higher risk of progression from a latent TB infection to active tuberculosis disease. So for example, the immune response in the elderly is characterized by this term called inflammaging, inflammation with aging. So as we age, we have a chronic low-grade inflammation. And that inflammation interferes with the control of growth of latent MTB. The elderly also have comorbidities, for example, a higher prevalence of diabetes or renal failure or cancer, all of which will compromise our immune competence. The elderly are also more likely to have damaged lungs because in previous years, smoking was a lot more prevalent. So smoking is more prevalent in the older individuals. And then the third reason is just knowledge gaps. The elderly are a neglected and understudied population. And in fact, when I started my studies in diabetes, I excluded the elderly because they would give me different results. So I'm not the only one to have done this, and now I fully regret it. So as a consequence, there are few studies in the elderly and very few in TB with Hispanics, and none in the Rio Grande Valley, except for the first publications that have come out recently from my group. So to address this knowledge gap, we conducted an epidemiological study of TB in the elderly from the Texas-Mexico border. And the design is cross-sectional. We enrolled elderly, defined as individuals 60 years or older, and adults defined as individuals 18 to 15-year-olds, and we enrolled a total of 646 participants. We enrolled community controls to enrich for TB-naive individuals and recent contacts of TB patients to enroll for people who have a latent MTB infection, and active TB disease to study people who have already developed TB. Our study sites were TB clinics in Texas, Hidalgo, and Cameron counties, and in Mexico, in Reynosa and Matamoros. Our working hypothesis was that diabetes is a risk factor for TB in the elderly. And why is this? Because diabetes is associated with a dysfunctional immune responses, as I have already shown you. Diabetes is a risk factor for TB in adults. So I have shown you that in adults, diabetes increases the risk for a latent TB infection and for progression to tuberculosis. And because the prevalence of diabetes increases with age. In addition to having a hypothesis, we also did a study where it's like a discovery type of study where we asked what other risk factors are there for active TB or for latent TB infection among the elderly. So we enrolled 744 participants, and we excluded individuals who were HIV positive, who reported excess alcohol consumption or drug abuse, because there were very few elderly with these characteristics. So the elderly probably die at a younger age. And so we ended up with community controls, 142, recent contacts of TB patients, 381, and pulmonary TB patients, 123. And so I'm going to tell you first briefly our findings among the non-TB patients, and then tell you about our findings with TB patients. So what did we find regarding latent TB infection in the elderly? So we defined latent TB infection as anyone positive for interferon gamma release assays. These are commercially available assays, and they probe our cell-mediated immunity to Mycobacterium tuberculosis, and they're the quantiferon gold in tube or the plus version, and the T-spot TB. And what we observe is in the younger people, there's about 30% of the younger individuals have a positive latent TB infection. So that's the background, latent TB infection in the community. However, as you get in the community to the older people, we found that more than half had a latent TB infection. So in the elderly, we have a reservoir of people with latent TB infection, and it is quite a huge reservoir. When we look at the recent contacts shown here in gray, it really doesn't matter the age. The prevalence of latent TB infection is pretty much the same. So what are the conclusions from these findings? So the first one is that these interferon gamma release assays appear suitable for the diagnosis of latent TB infection in the elderly. And the reason for saying that, I really don't have a gold standard, but as you increase in age, there's an increasing prevalence. This is in contrast to the tuberculin skin test, which is notorious for the false negatives in individuals with TB. The second is, what are the implications of a positive latent TB infection in an elderly individual? So if you're a recent contact, probably it'll be very important to have preventive treatment for TB because any recent infection plus old age will put you at a higher risk of TB. But among the community controls, we have quite a bit of people who have a latent TB infection. And I would argue, in contrast to what the literature says, that these individuals are even more resistant to TB. They are a selection of people who have survived. And I am looking forward to studying more these individuals at the biological basis level to understand what we can learn from them. So what have we learned about people with active tuberculosis in the elderly? So what we did is we sought to identify what are the risk factors for TB among the elderly. So we compared people with TB versus no TB and see what is the difference. So by univariable analysis, we found that in TB patients, there was a higher prevalence of male sex. There was more people who were smokers, and the TB patients were more likely to be underweight or to have a normal weight. So this is expected. This is what we see in adults who have tuberculosis. What were surprising findings were that impaired fasting glucose was inverse. We would expect that people with TB would have higher impaired fasting glucose, and we would expect them to also have more hyperglycemia because we have shown that diabetes is a risk factor for TB, but we didn't see a significant difference for hyperglycemia and an inverse relationship for impaired fasting glucose. Another very intriguing observation is that people who were BCG vaccinated were seemed to be protected from tuberculosis compared to those with TB. So we did a number of multivariable models to expand on these observations related to diabetes and BCG. So similar to adults, we found that people who are, so these are the elderly, the adults, these are the odds ratio, and any odds ratio above 1 in these models means that you have a higher risk for tuberculosis. So we found, just like similar to adults, anyone who is underweight or normal weight has a higher risk of TB. Males have a higher risk of TB, and people with a smoking history have a higher risk of TB. Again, to our surprise, even with multivariable models, we found that hyperglycemia or impaired fasting glucose were not significantly associated with TB in the elderly and nearly protective. In adults, we confirmed that diabetes or prediabetes is a risk factor for tuberculosis. And then for no BCG vaccination at birth, again, we find that it is a risk factor for tuberculosis in the elderly. And as the literature has shown again and again, BCG has no role for TB protection in the adults. So these findings are very recent, and we just published them, and now we're trying to understand what do they mean. And so, talking about BCG vaccination at birth and being protective versus TB in the elderly. So we know that BCG protects infants and children from disseminated TB. In fact, it is the most widely used vaccine worldwide. In years or decades, it's been known that children who are vaccinated with BCG will not only be protected from TB, they will be protected from other non-mycobacterial pathogens, so other pulmonary infections, diarrheal infections. But it is known that it does not protect adults from pulmonary TB. In recent years, Dr. Mihai Netea has really revolutionized science by showing what is the molecular mechanisms for these. BCG induces what's called trained immunity of monocytes and macrophage precursors. And this occurs in the bone marrow, and it can last for years. The question is, can that protective effect of BCG last for decades? So now we've got ongoing studies in our lab to evaluate trained immunity in monocytes, and we're just comparing individuals, elderly who have been BCG vaccinated versus no BCG vaccinated. So those studies are in progress. But an intriguing observation done by Belinda Medrano, a current PhD student. So Belinda is studying the elderly with TB. So she did a multivariable backwards regression model among TB patients. And she's looking for what are the host factors in TB patients that predict death from TB treatment. So as expected, she found that age is a huge factor, and I already had shown this previously, but a surprising observation again is BCG is coming up here again as protective for protecting TB patients from death. This is controlling, this is a multivariable model controlling for all these other factors. And although we cannot rule out other potential confounders, it's certainly an interesting observation. And then we're also following up on diabetes, a known risk factor for TB in adults. Why not in the elderly? So I've been breaking down the odds of the point estimates of the risk of diabetes in TB. And it is this is for all ages. And this is the younger people. And as we get older, the point estimates do go down. So there's something about older age. I also went back to data from colony forming units and the ability of mononuclear cells to contain mycobacterial growth. And I find that among people who are young adults, those with diabetes have a problem containing mycobacterial growth in vitro compared to people without diabetes. So this we know. But when I break it down into the older group, I just don't see any significant difference anymore between these two groups. Of course, I've got a smaller sample size. These studies were not designed for studies in the elderly, but we're replicating basically the epidemiological findings that we're seeing. So I think that there's something really interesting here. And so we're doing analysis of how is diabetes different in adults versus the elderly? How can differences influence TB risk? So we're taking adults and elderly with diabetes only and seeing what is the difference. So some of the differences that have called our attention are the history of diabetes. So the elderly are more likely to be aware that they have diabetes. The elderly are also more likely to be taking medications for diabetes, even though this difference was not significant. The elderly have had more years with diabetes and a higher glycemic index, but due to more chronic diabetes. Another observation from the elderly is that the elderly with diabetes have lower levels of fasting glucose and lower levels of HbA1c. So diabetes is more severe in the younger individuals. We've also done this analysis to visualize the difference in diabetes between the elderly and the younger people. And so in blue, we see diabetics who had both high HbA1c and high glucose levels. And you can see about 3/4 of them in the adults, but only half of them in the elderly. In the elderly, we had a high proportion of individuals who only had a high HbA1c. And in the elderly, we also have a high proportion who have everything normal, but they reported that they had diabetes. So we're looking into more hypotheses. As I said, this is a work in progress. And so the hypotheses are that maybe the elderly are not as susceptible to, the elderly with diabetes are not as susceptible to TB because they're taking more medications for diabetes, for example, metformin. And metformin has been suggested to have a beneficial effect, not just for lowering blood glucose, but also for controlling Mycobacterium tuberculosis. The elderly also have less severe hyperglycemia, and maybe you need to have a very severe hyperglycemia to really induce this low-grade inflammation, oxidative stress, and all these downstream consequences that lead to diabetes. So in summary, diabetes increases the risk of TB by 3-fold versus 20-fold in HIV, but at a population levels, diabetes can have a more significant contribution to TB. Therefore, every community should establish what's the population attributable fraction of diabetes to TB to determine how funds should be allocated at the local level. TB clinics now serve as hubs for new diabetes diagnosis for TB patients. And our most recent studies show that it is cost effective to extend this opportunistic screening to close contacts. The impact would be huge because I have shown you that earlier detection of diabetes will prevent complications, it's cost effective, it extends the lifespan and the quality of life. I've also shown you some data to suggest that SARS coronavirus 2 infection may synergize with diabetes to promote TB reactivation. Also, diabetes patients have defective mononuclear cell function that compromises their ability to kill MTB in vitro. So we have these tools in vitro, and we're now using it to understand the biological basis for the higher risk of TB in diabetes patients. And finally, the TB epidemiology in the elderly has unique aspects. So further studies in the elderly are warranted for this vulnerable and understudied population. And they're probably going to shed us light into the pathogenesis of TB in general. Now, this is my last slide. I just want to mention that the field side is also testing diagnostics pretty much all the time. And so we're now working on an application that I'm very excited about. And this is a method for detecting TB in rural areas. And this is developed by my colleague, Jordi Torreyes at the Texas Biomedical Research Institute. So what Jordi and colleagues did is that they designed a mixture of chemicals where you can put the sputum in this cup, and then you mix it for a few minutes, let it stand 10 minutes, and then you take drops from here and plate it in this agar that has a combination of antibiotics. Within 3 to 18 days, you're going to have growth, selective growth of Mycobacterium tuberculosis, and you're going to be able to identify not only multi-drug resistant mycobacteria, but extremely drug resistant mycobacteria. So I think this is very cool because you don't need a centrifuge. It takes a few minutes. These plates can be, if there's no electricity, you can even leave them at room temperature, and it will just take longer, but the mycobacteria will grow. So we're in the process of generating some preliminary data and writing a grant, and hopefully we'll get funded to do this and some other interesting studies. So with that, I want to acknowledge the people who have done the work. And Sue and Joe, I noticed that you have arrived. And Sue and Joe were my mentor during my postdoctoral training in early stages of my career, and I'm very thankful to that. In addition to that, I want to acknowledge my current team, Doris Ayala, Ana Paulina Quirino, Jose Caso Mateo, Sean Monday, Liz Calles, and I've described some of the work that some of them are doing, and then incredible field team, Genesis Aguillon and Julia Marlene Baena Olivares. I've also got a lot of thanks for all the people at the health department. This is 20 years of work and ongoing with a lot of very good collaborations, and I hope I've given credit to the people as I have gone along. And then I've received funding from NIID, the National Institutes of Health, and the U.S. South African Collaborative Award. I always like to finalize by saying that COVID-19 was a very, very tough year in 2020. I lost two very good friends, long-term collaborators, Dr. Francisco Mora Guzman, head of the TB clinic in Matamoros, and Emine Rodriguez Reina, who was my field person for the study site in Matamoros. So with that, I'll take any questions. Thank you.
Hello, Dr. Restrepo.
Hello.
Can you all hear me? Oh, okay.
Yes.
First of all, I just want to say that that was a great presentation. Thank you so much for that. I really learned a lot from the association between diabetes and tuberculosis today. So I wanted to ask if in the literature, or have you found any association between diabetes and complications of TB? Like, for example, like miliary TB or POTS disease or meningitis?
Yeah, that's a great question. And the answer is curiously no. So one of the curious observations about TB diabetes is that it is usually pulmonary. I thought at the beginning that we were going to see more extrapulmonary TB, as we see, for example, in HIV/AIDS. And in the case of diabetes, it's exactly the opposite. So I think what happens is in hosts that have a immunosuppressed or immunocompromised immune system, you may have dissemination. But when you have a host with TB, it's a dysregulated immune response. And so that may, in a way, help to contain the growth of the bacteria outside of the lung. But that's just my hypothesis. But that's a very curious observation and a very good question.
Hi, Blanca. This is Joe. Very nice presentation. Thank you very much. A lot of hard work. That's clear. So, Blanca, we've long had the discussion about the control of diabetes and its impact on TB. And is that part of your interpretation here in the sense that the elderly appear to have better control of TB? And do you think that, I'm sorry, of diabetes? And do you think that is part of the explanation of why you see this difference?
Thank you, Joe, and thank you once more to Sue and you. You were not at the first part of the presentation where I did some detailed of our work together. But yes, to answer your question, yes. So I think that the fact that diabetes in the elderly is not so intense it really helps. And in fact, it's very curious because it even seems to help. So a little bit of hyperglycemia may be even good because it facilitates the access of the immune system to the glucose, to access of glucose. So maybe a little bit of hyperglycemia may be even helpful. So I think that's part of the explanation. I don't think it's all the explanation. Right now, I'm running a whole bunch of models that I didn't have time to prepare today. But what I'm doing is I'm doing cutoffs. What if you've got elderly with high HbA1c? Are they still going to be equally susceptible? And it is not clear that that is the case. So I think that that is part of the explanation. But the underlying pathophysiology of diabetes in the elderly is different. In the elderly, there seems to be a delay in the release of insulin. And so there's not all this obesity, there's not all this other inflammation, there is a delay, there's not so much insulin resistance, there's just a delay in the release of the insulin. And so I'm trying to understand also, is it the insulin levels? Is there something else that is related? So I think there may be more than one explanation. And I think that these studies in the elderly are going to provide to us a new perspective to study and to understand tuberculosis pathogenesis from a new point of view.
I have a comment here, or a question really. One of the things we consistently observe is that the rates of diabetes go down in the elderly, and they're generally better controlled anyway. And this may be due to the fact that something else gets them first. They often don't survive to be elderly, particularly if they've got uncontrolled diabetes and a lot of cardiovascular and other complications. You know, they sometimes die even in their 40s and 50s. So I don't know what, we missed your definition of elderly, but by the time you get to 65, which is most people's, then we find that the curve really drops very sharply of the percentage of elderly who actually have diabetes.
Yeah, so that's a good point. And so what, even though I'm not doing a cohort and I'm not following them, so maybe that's something that you all could probably infer from the data you have since you're following people over time. But yes, my thought is that people who have very severe diabetes are going to die at a younger age. And so the elderly who have diabetes are survivors, and they are either they didn't have a very strong diabetes in their younger years, or they developed diabetes in their elderly years with a totally different mechanism, as I was explaining, a delay in that insulin release. However, what we do find is that the prevalence of diabetes and pre-diabetes, at least in the elderly that we enroll, is higher. So the prevalence of diabetes does increase. And we defined elderly as people who are 60 or older. So we went 5 years under the usual definition just because finding those elderly was difficult. But we find actually a higher prevalence of diabetes. It's just that it's a different diabetes. It's not the very acute high HbA1c levels so it's a different diabetes.
Sorry, Blanca, there's another issue that's an interesting one, and I think little is known. You may know more about it than I do, and that is the whole issue of the disconnect between HbA1c and blood glucose. And there are those who think this has that there is an important genetic component related to the glycosylation of hemoglobin, and that this may be why you see in some people it may be much more easily or glycosylated differently from others, and this may be why you see in some people, and it looked like you have a little bit of a disconnect in your older people, where you have high HbA1c but not high glucose.
Yes, that's a very interesting question. So really the definition of diabetes is high blood glucose, and HbA1c is an indirect measure. And HbA1c, as you're saying, can be masked by genetics, by anemia, by all these other factors. And in fact, when we did our studies with South Africa, in the colored people, South African colored people, we had a bigger disconnect. And what we found is HbA1c in that community, you get higher HbA1c. So you need to drop the cutoff like by 4%, for 0.4 points. In the elderly, though, we do see a disconnect. But what I think happens in the elderly is that you're seeing when we're doing our blood glucose, we're doing fasting blood glucose first thing in the morning. At that time, the elderly may have a normal blood glucose. However, once they have a meal, the blood glucose goes up and insulin release is delayed. And so the blood glucose stays up longer in the elderly. And this is what I think leads to a higher proportion of people who have a high glycated hemoglobin, but we're not detecting that hyperglycemia in their fasting glucose. In fact, that for detection of diabetes in the elderly, it is recommended that you do an oral glucose tolerance test, because you're going to miss 30% or even more of the diabetics in the elderly years because of that. So I think the disconnect we're seeing may be a real, the HbA1c may be telling us the other half of the story that we're not detecting with the fasting blood glucose. But I really don't know. It's just based on the pathophysiology that I'm speculating that.
I want to give other people a chance to ask questions. Blanca, but
Sure. But those are great questions. Thank you.
anybody else have a question?
Blanca, can I ask a quick question? This is Maria. It was
Sure.
really great presentation. I learned a lot. Two questions actually. One, that when you do the modeling with the enhanced study, you do not have an ethnicity or race variable in the model. Have you tried that one?
Yes, we did. So there, because of the lack of time, I didn't go into the details of the models, but we tried race, ethnicity, and it didn't have an impact. However, we find that when we do only Hispanics, the model may shift a little in terms of its behavior. And it may be even more sensitive in the Hispanics. So yes, there's an influence, but when we selected our final model, race ethnicity was not a strong variable, so we excluded that from the model.
Okay, thank you.
Sure.
And second one was basically when you do the Markov chain analysis, that was really interesting simulation. And how your co-authors incorporated age in terms of the quality? Because it is quite related variable, right? Quality. Yeah.
Yeah, so the short answer is I don't know, and I don't think that Robbie is here. And so I really have to defer to Shelton and Robbie, who are the mathematicians. And I'm just learning a lot, but I don't understand all the details. But basically, I think Robbie started the model at age, I believe it was 45 or 35. And then
Okay.
a follow-up over a 45-year period of what are the probabilities of each of the diabetes complications. And so I know that Robbie played with different ages. This is the this is like the final model, but he's got a super cool Excel sheet where you can change sex ratios, you can change age, you can change everything. And the nice thing about the model is no matter how he did the analysis and what variables he included, it was always clearly cost efficient to do the screening for diabetes, and there was an expansion, a lifespan, and a reduction of complications. So it seems like a very robust, and it sounds like a no-brainer. But I hope that with these models that I presented, I can convince the rest of the world that we should be having the contact screen for diabetes. I think this is important.
Okay, great. Thank you so much.
Sure.
I'm going to give people another chance to ask questions. I got one for you, but I want to make sure that others have an opportunity. Okay, so I wanted to first off, thank you so much for a wonderful presentation, and really for just framing your 20-year career and what you've learned in a 1-hour presentation. That alone is heroic. I have always admired the way you have a laboratory element to your research, a field element to your research, and then take it to the public health sites and use the research. Thus, I'm explaining, right, a strong translational component to your research. And not many researchers actually do that and do it as well as you have done it, and will continue to do it, no doubt. But I wondered if you would just kind of give us some advice on what you've learned again over those 20 years in setting that up, in some of the ups and the downs of that type of work. I wonder if you would present some of your insights on that.
Thank you, Belinda, for those words, and thank you for your support as current dean. And it's certainly not easy. Perhaps one of the biggest challenges has been how everything changed on the Mexican side of the border. So we had set up a system where we would have a driver who would go collect the specimens from Mexico, bring them back. And then all of that was shut down due to the local public health situation. And so for many years, that was not possible. And then what I would say is just think outside the box. And then I thought, well, why don't we do the opposite? Why don't we have Mexican nationals bringing the specimens to us? And it was not easy. It sounds very simple, but I had to discuss with the lawyers in Houston, how are we paying them? And the university in Houston and the lawyers are telling me that that's a liability to the university. And I'm arguing, what is our goal? of being in the border, if we cannot do studies across the border. And I told them at some point, they were giving me all kinds of blocks for this to happen. And at some point I told them, okay, so you tell me, what do you propose? And there may not be a perfect solution, but let's reach some sort of agreement. And that totally changed the tone of the conversation with the lawyers. So I think that's one example of how maybe when you're being blocked, but you know this is important, you can shift it around and have that happen. And then another huge situation where I thought this was the end of it was when Trump, no politics brought, but so Trump made the border a lot more stringent. And we were forced that we had to bring our specimens through a formal importation through the truck lanes. And our blood specimens were arriving very late in the day. The blood specimens were not good anymore. And I've just contacted everybody you can think of, politicians, everything. And we've set up a system today where I communicate on a daily basis with the bridge to coordinate the entry of the specimens. So those are just two stories to say, having set up a site, I probably have access to more fresh TB specimens in mainland US than any other place in the US. But it's not easy. It's a constant, kinds of things come up. So I'm solving situations on a frequent basis. But here we are, it's still working, and it's generating little by little a lot of interest from a number of collaborators. I didn't have time to go into all the other stuff that we're doing. I just selected some themes. But thank you, Belinda.
It's a great example of the untold story, right? There's the publications and the grants and patient care improvement and your career advancement, but the behind the scenes, how does it really happen? So thank you for sharing that. Guess I should do one more call.
I wanna really, one of the things when I was having these troubles and Joe was dean, I had a conversation with Joe and he suggested how I could travel to Mexico and get a special permission and everything. So Joe, that was very helpful. That was one of the things that helped me move these ideas forward and make it, how do we get this happen and show my face in Mexico? And because you have to show your face, it all doesn't happen remotely.
Yeah, that was helpful, Blanca. I'm still on that committee actually that decides about travel. So I was able to twist a few arms and get special permission for you to continue to go across. Blanca, there's one other thought I just had, and that is that you have an admixed population because all of the Mexican population is admixed in terms of European and Amerindian ancestry markers. And one of the things, if you recall, a long time ago when Weijie Chu was here, we looked at insulin resistance in our cohort related to the percentage of one type or other of predominance, if you will, of either Amerindian or European genes. And those, when you have a predominance of Amerindian genes, you have a higher insulin resistance level. So you might, if you could genotype your at least enough to get ancestry markers, that's another variable that you might look at to see if there's any relationship there, either with glycosylation. We have not looked at that. It made me think about it. Well, we should go back and look at that. But also there may be some other factors here that we don't understand.
Yeah, that's a good point. And I've always kept DNA from all the participants and signed informed consents where there's permission for future DNA studies. So I think that's a very interesting idea.
Any last questions for Dr. Restrepo? Right. Thank you so much for the wonderful presentation. I think we should give her, you can give her virtual rounds of applause. There they go.
Thank you.
Great.
Thank you for the opportunity and thanks a lot.
Wonderful. All right.
They're
And
coming.
keep coming around. That's right. We love it. All right. Thank you so much, Blanca.
Okay. Bye, everybody.